Today we will be teaching you about DNA replication.
The parental DNA is semi-conserved when it goes through replication. The parental DNA goes through DNA topoisomerase, which unwinds it from a helix. It's separated into two strands, a leading strand and a lagging strand. The leading strand is in the 3 to 5 direction, and the lagging strand runs in the 5 to 3 direction.
DNA topoisomerase. DNA topoisomerase is an enzyme that unwinds the DNA helix. Although there are two types of DNA topoisomerase, only DNA topoisomerase 2 can break up DNA strands.
Helicase. DNA helicases separate double-stranded DNA into single strands, which allow each strand to be copied. DNA polymerase 3. The DNA polymerase 3 is an enzyme that joins covalent bonds between the nucleotides and the new strand, joined to the leading strand.
Primase. Primase catalyzes the synthesis of the short RNA segment called a primer.
DNA polymerase 1. DNA polymerase 1 fills in the necessary nucleotides between the Okazaki fragments in a 5 to 3 direction, proofreading for mistakes as it goes. DNA ligase, a type of enzyme that bonds Okazaki fragments on the lagging strand together. Now we've gone over all the steps of DNA replication. From here, we're going to go over how they fit together. The parental DNA first goes through DNA topoisomerase, which unwinds the DNA helix. From here, the parental DNA goes through the replication fork. It is then split into two separate strands, known as the leading strand and the lagging strand. The leading strand is created in a 3 to 5 direction by DNA polymerase 3, created towards the replication fork, which is opposed to the lagging strand, which goes through an entirely separate process. The lagging strand is run in the 5 to 3 direction, which means DNA polymerase 3 cannot work on it at the start. DNA primase then puts down RNA primer onto the lagging strand, which is exchanged by DNA polymerase 1 into DNA nucleotides. These DNA nucleotides are then put into Okazaki fragments by DNA polymerase 3, and ligase then comes and combines them together to form the remainder of the strand. Thank you.